Sorry. Short contents. I want to say some words firstly to the Institute. Then I want to show our Aptamer generation process, how it works in our institutes. And then in the end, I will give you some examples and project where we was working at the moment. So firstly to the Institute. So our institute is integrated in the Fraunhofer Society. It's a very big society in Germany. We have also some locations international, but they are working over 30,000 people in 76 institutes, and we have a research volume of 3 billion euro per year. And you see here the location of the institute in Germany, so it's very distributed in the country. And we have several topics where we're working on that. And our institute, of course, is located in the life science and health part. And it's at the moment 9 institutes working in this area. Our institutes have 100 people working there inside. We have a budget of 9 million euro per year, and we have 4 divisions in our institutes. One is located, it's working on automation, process automation, and the other one is a big one, is making here in molecular and cellular analysis, and another is bioproduction, where is located our group. And so we working in the cell-free system particular, but also in developed Aptamers. What we are doing in our working group is the core business is to generation of Aptamers, so DNA or RNA. We have an 4-phase approach for that, and have an automatic system, and we have also a management system to get a high success rate. Then we synthesize the aptamers, of course, for stabilization is one case, and also for functionalization. So to bring it on a surface of a sensor, or to couple the dye for a kind of assay. And of course, then we want to transfer it in applications, and that can be analytical Aptamer application, but also therapeutic applications. So it's a core business of Fraunhofer. It's a hybrid of a research institute or of a company, and to bring research, basic research in an application. So next, I want to go to the Aptamer generation process. So mostly of you know this process. You start with the target, and you have the individual selection, the sequence analysis, and the characterization. Then you can make some modifications and optimization until you have the Aptamer in the end. And we offered a full service for the generation of Aptamers. It's also divided in this 4 process. And so you can come to us, and we make a full subcontract in Aptamer for you, an RNA or DNA Aptamer. And it really works. From the starting point, the library, we have heard yesterday some information about Greg about this. We have also this primer constant, and we use different libraries in the range of 15 to 18 nucleotides inside. And of course, normally you use only 10 to 14 to 10 to 17 of diversity of every library you use, so 1 to 100 nanomolar. And I like this mathematics excurse to see the full sequence realm that you have when you use 40 nucleotides for randomization. It's 10 to 24, but would be when you would be realized that every sequence is in the starting pool, then you need 2 moles or 50 kilogram, and of course it's not possible to start with that, and that's the reason why you have this reduced amount. So you know this standard ZELEX process. What we have done is that we have make an automation of that. So that is based on magnetic particles that we use. So we couple the target molecule to the magnetic particles, and then make the incubation step and washing and dilution step in the robot system. And we use for that the biosprint or Kingfisher, so that we transfer the beads and not working on with a batch procedure where the beads were bind on the batch and you transfer the solution. And in the case when, so normally this we use with proteins or cells, but also we can, when we work with small molecules, we take this capture the Lex process. So that is a change of the arrangement. So you couple the library to the beads and the target is free in solution. But in principle, it's the same process. What we also do, we management this process. So we look at every selection round to the affinity. Of course, we use PCR, but we use also a fluorescence binding assay for that. And we looked also for the diversity. So it's also a very important point that you look to the diversity, that you know if there's a possibility to get a high enrichment or not. Then the next, of course, is a sequence analysis. And of course, we use NGS sequencing that everybody uses nowadays, I think. And when we make a bioinformatic analysis of the Aptamers, and for that we use, somebody knows this company, Apta IT, we use this software for that, and it works very well for analysis. So we can also do this in a service contract if you're interested in that. And then, of course, we like to characterize the aptamer, and we look to the affinity, of course, but we also want to look to the specificities, very important to compare that. And we use different technologies for that. So we have a fluorescence dye assay, and then really use, of course, electromobility shift assay when we work with protein mostly. And we have also microscale thermophoresis and SPR. And I think we have seen in the last 2 days many examples for that everybody uses for characterization of the aptamers. And then I come to 3 examples that I will show you where you're working on which topics at the moment. And let's cannot see it. This work is doing by Yannick Kerler. He has presented yesterday also a poster. And this is an aptamer for detection of glycogenization pattern. So you have various glycogenization pattern in autoimmune diseases, for example, rheumatic arthritis. It's very famous for that, and you have a decolonization, galactosylation of the N-glucans to G0 type. You can see it, and that it's connected with an increasing of the autoimmunity and pro-inflammatory effects. And so the G0 glucans is a good biomarker for these diseases, and normally it's a very cost and time intensive procedure for analysis, and you need normally lab personnel for doing that. And so we want to develop a rapid test, and that is firstly the generation of an aptamer against the G0 type N-glucane, and also this detection type what we want to develop. We want to use this aptamer is coupled with two fluorophores and a quencher, and when the target is bind, you get a confirmation chains, and then you get a signal in the end. For further information, you can visit the poster of Yannick. He's also in the audience. The next project where we are involved is with a company together. It's Nal van Minden, so somebody knows him. He's also visited this meeting in the past. And that is done by Wiebke Zabrowski. So you cannot show her faces, but sorry. It's going on the, so everybody knows the reduction of antibiotic resistance in bacteria. It's a very famous topic. And the reason is that there's a massive use of antibiotics in human and animals that you get this resistance. And so particularly in gram-negative bacteria is very critical. And so you need a rapid specific detection of the right bacteria to give specific and not a broad spectrum antibiotic for the people who fall in these diseases. For that case, interesting is the beta-galactam antibiotics. So it's a famous group because there is it's called like an antibiotic of the last resort because they have resistance against antibiotics. They are resistant against the beta-lactamases. And so far, rapid detection of this was very helpful to prevent this antibiotic resistance in future. And for that case, we decided to make Aptamer against the carbapenases. And the carbapenases are very famous, very different carbapenems in enterobacteriales. And we used 3 proteins from this class for Aptamer selection. And we were successful in all 3 selection processes and get Aptamers against these 3 types. And we have also developed with the company together a lateral flow assay for that. And we developed as a competitive assay, so working with 1 aptamer or with a sandwich format assay, so working with 2 aptamers or with a combination of an aptamer and an antibody. And unfortunately, I cannot show the results in detail because we have made the publication, it's submitted and it's in revision at the moment, but it's not accepted. So perhaps next meeting I can show the very good results in detail. And the last topic is a diagnostic in bladder cancer. It's another high topic in health. And that is the case is when you have bladder cancer, it's one of the 5th frequent cancer. And the gold standard for the diagnostic is a cytoscopy or a biopsy. And the biopsy, it's not so nice when it was making with you. And so there is an interest to have a rapid detection for helping, do you need a biopsy or not? And for that case, we decided to make a rapid detection assay, an aptamer-based assay. And so we looked for biomarkers for the bladder cancer and want to develop then also a lateral flow or microtiter plate assay. And we found then that a good biomarker for this bladder cancer is the urokinase type plasminogen activator, UPA. And this is a protein that make an autoconversation in 2 forms, the high molecular weight and the low molecular weight. This work is done by Nico Dreimann. And we have also here, and you see it here, this is the full protein, and then the low molecular weight protein is in blue. It's only this catalytic domains inside. So we have a catalytic domain in this protein and a receptor binding domain, the RD. And we have developed several aptamers against this UPA form, against both UPA forms. And we have also aptamers that binds to different epitopes. And that is very nicely in that case. So that we have used this for development of different sandwich assays. So we have developed aptamer sandwich assay, very successful, where with 1 aptamer is then coupled over biotinylation modification on the surface, and the detection aptamer has Cy5 label that we heard in the last talk, similar. And we have also tested combinations of aptamer antibody or antibody aptamer sandwich. That means that the aptamer is the detection or the capture molecule. And it works also very well, and it's the same case that I showed it before. Sorry for that. That also the paper is in revision at the moment, and hopefully next month you can read both in public. But what is very interesting in this case that I will mention in the end, this aptamers has a potential as therapeutical agents, because one aptamer inhibit the receptor binding position, and the other aptamer can inhibit the catalytic activity of the protein. We have tested that, and yeah, we will publish that in the paper. That's very, very interesting for also for detection and also for therapeutical of this bladder cancer. And I will also mention that UPA is also a biomarker for other cancers, especially for breast cancer. So it has a potential, this aptamer, to use in other cancer detection. Yeah, I will thanks in the end to the department in our institute, so not my group. It's a little bit smaller. The people are mentioned here, and I have a smaller picture of the group from the last Christmas event. And in the end, thanks for your attention.